Thank you, Dr. Sindhura and Samshir for inviting me here. It's indeed a great pleasure to be here amongst all of you this afternoon. So the topic is overcoming challenges in embryo development and implantation failure. So to define the scope of this presentation, actually I have split it into two halves. We would be looking at the developmental arrest first and then move on to implantation failure next. So what is developmental arrest? We'll start with that. We will also look at the factors which are involved: maternal, paternal, genetic, metabolic and cultural conditions. And then we will go to implantation failure and we'll see what are the factors involved in implantation failure. And I'll also take you through the good laboratory practices to overcome certain of these problems. And finally, we will sum it up. When do we call it's a developmental arrest? When you see that, like the embryo does not have any cleaving activity for 24 hours, there is no mitotic cellular division for at least, like one full day, that is when you call it that embryo is arrested. Now, what is the incidence? 30 to 50% of the embryos only reach blastocyst stage, meaning the remaining 50%, they get arrested at some point or the other. And more than 40% of the patients undergoing IVF have at least one arrested embryo. So that is the incidence which is reported in the literature. Now, what are the major reasons for developmental failure of embryos? It could be intrinsic. When I say intrinsic, it could be maternal, it could be a paternal origin, or it could be some metabolic activity which is not there in the embryo. And then crucial molecular pathways could have been affected. All these, and genetic abnormalities. And extrinsic, it is something what we do in the laboratory, the protocols, the culture conditions, so on and so forth. Now, let me start with oocyte maturity. I have started it with this particular point because most of the time what the doctors or the clinicians, they would say is, I gave you so many M2s. Like I gave some 20 M2s, but I got only 3 blastocysts. All of them have, the others have got arrested. But here we have to see this mature oocytes we are talking about. There are two aspects of it. One which is nuclear maturity, the other one which is cytoplasmic maturity. When we are saying nuclear maturity, the picture here actually tells you that you see a germinal vesicle. This is an immature oocyte. This germinal vesicle actually breaks down. It becomes a M1 oocyte, which is on the way to become maturity. And here you have a mature oocyte with a polar body. So this is something which you observe under the microscope and you say that the oocyte is mature. However, there is another aspect which is the cytoplasmic maturity. See this, the cytoplasm here, it undergoes a lot of changes during the process of maturation because there are a lot of organelles there, and these organelles, they get kind of spatially distributed, which actually plays an important role, and that cannot be visualized under the microscope. I have shown you a fluorescent microscope image here. See the distribution of mitochondria in a GV oocyte. Now, when the GV breaks down, you see the same oocyte will have its mitochondria getting, like organized in a certain way. Now, this is a mature oocyte and you see the organization of mitochondria. So, can you see the difference? This will not be visualized under a stereo zoom microscope. So, even though we say it is an M2 oocyte, we may not have these things in the proper place, and that actually could be the reason why the oocyte does not grow into a blastocyst. It might get arrested at some point or the other. Now, this is a study. It's an interesting one here. Actually, this, what they have done is they have just segregated the patients depending upon the proportion of immature oocytes. And you can see here the first group, the group 1 has 15% of immature, less than 15%. Group 2 has 16 to 25%. And group 3 has 26 to 50% of the retrieved oocytes were immature. And group 4, the percentage of immature oocytes were more than 50%. Now they just looked at all these parameters. But see, this is of interest. The ET cancellation rate due to poor embryo quality was much higher in this group. Can you see this? Like, so whenever there is a percentage of immature, higher percentage of immature oocytes in the number of oocytes we retrieve, then, the embryo arrest is more. So, like what happens is you retrieve some 20 oocytes. And out of that, 10 are immature, 10 are mature. But then, even then, you might have got 10 M2 oocytes, but still the cohort is mostly immature oocytes. You can actually expect a lot of cleavage arrest in this group of embryos. Now, the next one is oocyte morphology. This is a no-brainer because when you have abnormal oocyte, definitely it will have an impact on the developmental potential of the oocyte. And this is a study, again, an interesting one, where they just looked at these parameters like granular cytoplasm, fragmented polar body. So with these two parameters, they actually found that the developmental arrest is much higher. So this is what we call a granular oocyte, and this is what we call fragmented polar body here. Not only the morphology of the oocyte, the consistency of the ooplasm also matters, because when you do ICSI, when there is a sudden and difficult ooplasm aspiration, those oocytes definitely have a higher probability of ending up in developmental arrest. Now, so this is something which you can visualize under the microscope. But as I said earlier, there are a lot of organelles which you cannot actually see under the microscope. And one of them is the maternal mRNA. And maternal mRNA is known to support a lot of different functions regarding oocyte maturation and fertilization. So why am I even saying this? Because it is shown that maternal mRNA is affected in oocytes from smaller follicles. So that is the takeaway here. See, sometimes what happens is the follicle size could be less than 12 mm, but if you give HCG, you will still get an M2 oocyte. But because it is coming from a smaller follicle, the maternal mRNA is affected, and those oocytes, they have, lot of their functional activities disrupted, and they go in for early cleavage arrest. So keep in mind, when you are looking for developmental arrest in your lab, just check the follicular size also on the day of trigger. Now, what about the paternal effect? Definitely that also has an impact. Now, this was a study where they just looked at 3 groups of patients with a total motile sperm count less than 1, between 1 and 3, and then total motile sperm count between 2 and 3 and 5 million. And they found that the embryo developmental arrest was 24% in group 1, where it is less than 1 million, and it is comparatively lesser in group 3, which is more than 5 million total motile sperm count, and this difference was found to be statistically significant. And then when you do TISA, again, if the sperm is from testicular origin, then you still have a chance of embryos arresting before the blastocyst stage. Not just the sperm parameters, the paternal age also has an impact. And this was a study which was published in 2008. They found that donor oocytes were used. So you can kind of rule out the oocyte quality here. And they found that the embryo arrest and at the cleavage stage was significantly lower in men less than 50 years of old. So as male age increases, there are significantly more chances of embryos getting arrested. And what about the other reasons? Like genetic reasons are there, like Dr. Aruna had spoken about genetics and immunology has a significant role. This is something which we do not see in the laboratory. Under a stereo zoom microscope or even an inverted microscope, this is not visible, but however, they have a major role in taking an embryo to become a live baby. Now, a lot of genes are involved. So I don't want to go through the whole thing here, but what I want to say is the correlation between mitochondrial DNA and the genomic DNA has a role, and then the correlation also between the mitochondrial DNA and the nuclear DNA also plays a very important role. So when you have developmental arrest, when all your other parameters are controlled, and this is what actually you have to look at the genetic reasons. Now, are genetic reasons only it is very easy, right? Like for the embryologist to say that, no, there could be some genetic reason, that is why we are not getting blastocyst. But that is not the only reason. We can also have other reasons such as metabolic reasons. See here, this is another interesting study where they found that even euploid embryos, they get arrested. So if you are saying, genetic reasons are the reason for developmental arrest, then all euploid embryos should actually become blastocysts. So here the testing was done on day 3, and they found that day 3 euploid embryos, majority of them, they did not, 63% were arrested before it became blastocyst stage. So, like there could be factors. And one of the factors which is mentioned is metabolism. And these studies were done long before in the 90s. And Gardner's group was one of the major group involved in metabolic activities of embryos. They found that certain factors like pyruvate, glucose, the uptake and the utilization by the embryos is very different between an active embryo and an embryo which is going to be or which is destined to die. And this is one of the recent studies, and here they just some 201 peptides, which is coming from 107 precursor proteins. They are involved in biological process, and they are significantly different between viable embryos and the non-viable embryos. Now, reactive oxygen species. There was a time that whatever is happening to a human body, it was blamed on reactive oxygen species. Like even cancer was said that its major reason is the generation of reactive oxygen species and lipid peroxidation. And in IVF also it is there, but however, there is contradictory evidence because, see, the main reason is it's very, very difficult to quantify reactive oxygen species. However, embryologists should keep in mind that whatever they do in the lab can actually trigger reactive oxygen species production because the components in certain commercial media actually can create reactive oxygen species in the culture environment. And this is one thing which, I want people to concentrate, the oxygen concentration of culture. If you are using those box type of incubators, which is not controlled for oxygen, where you use ambient oxygen, 20% oxygen, then definitely you are increasing the chances of reactive oxygen production in the culture system. Visible light, how much do you expose? If you visit the European labs, you actually see they are working in practical darkness. You even think that how are they even able to see anything inside the lab? That is the level of low light in the labs. I'm sure, it is difficult for us to get acclimatized to such a work environment. But however, in the laboratory, if you can actually minimize the visible light, that will definitely help. And centrifugation. When you are centrifuging samples, you have to centrifuge it at a particular RPM or particular G-force. But if you just, to make the process easy and quick, if you just run it through a very high centrifugal force, then you are generating reactive oxygen species in the sample, and that sperm, if it is used for insemination, it can also cause developmental arrest in the embryos. This is just quick some data from NOVA, and we looked at close to 50,000 OPU cycles done in the last 3-4 years. And you can see here, first we kind of segregated amongst women who are less than 34 years, between 34 to 38 years and 39 years and above. We just looked at the developmental arrest. What is the percentage of embryos getting arrested? And you can see in women who are less than 34 years, the embryo arrest is 5%, whereas in women over 39 years, it is 15%. So, like with age, definitely you have a lot more chances of embryos getting arrested. We also looked at the women where we actually retrieved less than 4 oocytes and compared it to women where the number of oocytes retrieved were more than 4. This again, we looked at some 50,000 cycles from the last 2-3 years. And as you see here, the percentage of embryo arrest in women, in poor prognosis women, where we retrieve less than 4 oocytes, it is 17%. And then in women where we retrieve more than 4 oocytes, it is significantly lower. All this data is statistically significant. Then we also looked at the sperm quality. We compared it between normal, abnormal, and borderline. And the developmental arrest in abnormal sperm, when we use abnormal sperm, it's 8%. And then when we use normal sperm, it is 8, and then when we say borderline, this could be some analysis issue, but these values are not statistically significant. But then please understand from this, see, we are not saying that it is like 100% of the embryos get arrested. That is the beauty of IVF. You cannot say that if you do this procedure or with these conditions, you will not get any pregnancy. You only have to look at the average and see that comparatively, you might get a lesser number or a higher number of whatever parameter you are looking at. Now, moving on to the next one, that is the implantation failure. We know that like how much ever good embryos we transfer, still implantation failure is a problem in IVF. And for successful implantation, we need a competent embryo and we also need receptive endometrium. And embryo competence, it accounts for 60 to 70% of implantation failures. Now, whenever we have implantation failure, two things we have to look. Did we transfer good quality embryo? Or did the embryo quality was poor? If the embryo quality is good, now before we even go into that, there is something like I would want to spend 2 minutes on. How do you even define an embryo quality? Because when you talk to embryologists, they would say that, now embryo quality was very good. No, but then there are certain strict parameters. Let us start with, like it has to be a good pronuclei. It has to be a good cleavage stage embryo. It also need to be a good blastocyst. That is what would define a good quality embryo as. Now that brings me to another question. Is fertilization check necessary? The reason I'm asking this is in many centers where they just depend on freelance embryologists, they say that it is not necessary to do fertilization check. And the reason they give is, you are unnecessarily exposing the embryo to ambient conditions, so you don't have to do it. But I am a very, very strong proponent for fertilization check because that can actually give us a lot of information. This is the summary of the information which a fertilization check can give. Please, you can just take a note of it. Since we do not have time, I don't want to go through the whole thing here. But a proper fertilization check done will give you an idea whether this embryo, I'm not saying 100%, but at least a major percentage, you would know that this embryo is going to go to blastocyst or is it going to get arrested. But then remember, fertilization check has to be done at 17 hours plus or minus 1 or 2 hours. Like we cannot do it at 20 or we cannot do it at 13 hours and say that pronuclei are not good. Similarly, there are some people who would just do ICSI and then they will only take the dish, and if it is a single step media, they will take the dish on day 5. No, we also need to look at either a day 2 embryo or a day 3 embryo. If you are doing day 2 assessment, it has to be done at 44 plus or minus 1 hour post insemination, or 68 hours post-insemination, post-ICSI. And then you have to have a very defined quality mentioned there, how many cells, what is the percentage of fragmentation, so on and so forth. Now, when you say a stage-specific cell size, see here, this blastomere is slightly bigger than the other one. Then you also see the cell-to-cell contact between the embryos. Definitely, if you show this picture to an embryologist and ask, is this a good embryo? The embryologist would definitely say that, yeah, it is good because blastomeres there are no fragmentation. But however, you see the difference between these two. There is a good cell-to-cell contact here. So, like this is a better embryo. And these kind of assessment has to be made by the embryologist when they are checking. That would definitely help us to understand either developmental arrest or implantation failure. Again, the spatial arrangement. See, this is also a good day 2 embryo. But can you see the arrangement here, the clover-shaped arrangement? This is how a day 2 embryo should look like. Because, see, all these things matter because when cell divides, there should be equal distribution of the nuclei and other organelles between the cells. But if they are like this, that pattern is affected and that actually would result in either implantation failure or even before that as a developmental arrest. Now, blastocysts also have to be scored. So, a good embryo, again, we just don't say good, bad, ugly. We have to say, what stage of development the embryo is, what is the grade of the ICM, what is the grade of trophectodome. So, it should be actually, like very well defined, and there should be a systematic way of looking at embryos. This is again another question. So, what is the implantation rate between a day 3 transfer and a day 5 transfer? Most of the studies are actually favoring day 5 transfer, and the chances of cancellation of embryo transfer is higher if you are waiting to be, the embryos to go to day 5. But if you are ready for that, if you are ready to counsel the patient, I think taking all your embryos to day 5 will give you a significantly increased success rate and significantly increased implantation in the lab. Now, even when you get blastocyst, sometimes the blastocysts do not hatch. I had the opportunity to work with a time-lapse microscope, and then from there we knew that it's a beautiful blastocyst. But then what happens is expansion does not happen much, and then there is no hatching also. And even in the literature, it says that, close to 20% of the expanded blastocyst will not hatch out. If they don't hatch, then definitely what happens is you put it in the uterus, but still it will not implant. These are the things when you're just looking at a good embryo, but when you have a poor embryo quality, what do you see? Dr. Sarapreet had already spoken about all these parameters, like we have to look at the air quality, we have to look at the culture system, the temperature, and it has to be measured. You cannot, outsource it to somebody who comes and measures once in 15 days or once in a month. There has to be a temperature meter in the lab where you have to do it diligently every morning. You also have to measure pH. Osmolarity you cannot measure, but however, do not do things which can compromise osmolarity. On all your equipment has to be serviced, and then the service reports have to be filed in properly so that the culture conditions are not compromised. And culture media and consumables also, has to be carefully sourced and carefully stored, carefully used, so that, it will not affect your growth of the embryos, nor it will affect the implantation. And manpower, I don't have to say that with ART, we have very, very strict criteria of hiring embryologists, and it is necessary because sometimes what happens, embryologists look at people working over the shoulders, and then they just think that they can do, and then they go to the labs and start working, and you can't give results. Like any medical professional, I think there has to be a formal, if there is no formal education, at least there should be a formal training in these procedures, which will have an impact on the outcome. Now, these are some of the critical parameters. Again, like when you have abnormality in temperature, what happens? The spindle will get disturbed. And then when the pH is not maintained, then the cytoplasmic granularity increases. Now, when you have VOC exposure, then this will cause increased DNA fragmentation and mitochondrial stress. Then when the media composition is compromised and you have altered methylation in the embryo, then when osmolality is compromised, then you have poor compaction and zona hardening, there will not be any hatching. And then if you constantly expose the embryos to bright light, that can actually cause spindle damage and fragmentation. So if you don't maintain your culture conditions well, definitely the embryos will not grow further. And even if it becomes a blastocyst, there is a saying that it might take a last gasp at the blastocyst stage and die. So, there will not be any implantation. Now, what are the best practice recommendations? I have just compiled a few things here. As far as possible, try to use tri-gas incubators. When minimize dish exposure and monitoring with activated carbon, your filters should have activated carbon. Quality control calibration is must, and as I said, do not outsource it to people. Just have all these equipment in your lab and you do it diligently. Have incubators depending upon the number of cases you do. If you have less number of cases, have work with 2 incubators, but with the caseload increasing, you have to have more incubators. Then media has to be validated, equipment has to be optimized, and try to get experienced and skillful embryologists. Now, embryo transfer is also important. There are, lot of, good embryos, but no implantation. You also have to look at the transfer, not just the transfer skill of the clinician, but the transfer skill of the embryologist also has to be assessed. What is the volume? How much of air is there inside the catheter? All these parameters may play a very important role. So to sum up, developmental arrest and implantation failure can be due to multiple causes. Though genetic factors are one of the major cause, age is also another important cause, and maternal factors are involved. But then as embryologists, let us concentrate more on the lab conditions because, you are trying to mimic what is happening in nature. So you have to pay that kind of an attention to the growth of embryologists. Please remember that stable culture is equal to stable outcome, and embryo quality assessment and selection is very important. I have seen embryologists, the consultants would be upset. So they would just say, oh, I have a very good embryo, because they think that anyway, we are going to do frozen embryo transfer. I still have 3 more weeks. And then if it becomes positive, it is fine. If it becomes negative, I'll somehow manage it at that time.
So, but then please don't do that. Like, see whatever you are actually seeing there and write it there in your recorded there. And embryo loading and transfer techniques also have to be optimal. So if you have all your culture conditions and parameters in a very controlled and consistent way, then we call ART an art, right? Like, it will no longer be an art, it will be a reproducible science. There will be some predictability in whatever you are doing. Otherwise, like, you just transfer and then you are waiting with your bated breath for another 15 days, not knowing, whether this is going to be successful or not. They'll be very confident. And if it doesn't work, there could be reasons. But if you can see, like, they'll say, I expected this embryo to be successful. So that is the kind of reproducibility we have to have in embryology labs. So with that, thank you very much for your kind attention.